this lecture, we would be studying about the basic concepts of enzyme. What are enzyme and what are their nature, how they are highly specific, what are the substrate, what is the substrate specificity, what is the coenzyme, what are the cofactor, what are the apoenzyme, what are the holoenzyme, what are the ribozyme, and what are the isozyme. So actually enzyme are biological catalysts. They catalyze a biochemical reaction, and they enzyme catalyze a biochemical reaction by lowering the activation energy. And actually how enzyme catalyze the biological reaction and how enzyme catalyze a biochemical reaction and lower the activation energy. Enzyme actually is having a site where substrate can bind, and we say this site is a active site composed of the two side. One is the binding site, one is the catalytic site. So the site where substrate bind is called the active site. It is called the binding site. However, the actual site where actual biochemical reaction takes place is called the catalytic site. So enzyme all those enzyme, they are having they catalyze the same chemical reaction, but they are having different amino acid in their polypeptide chain are called as isozyme. So isozyme, all the isozyme catalyze the same reaction, but they are slightly differ in the sequence of the amino acid in their polypeptide chain. All those enzyme may be non-protein in nature. We call as a ribozyme. Ribozyme are actually RNA molecule that catalyze a biochemical reaction. So enzyme could not be able to work without the help of other molecule. So the other molecule that are non-enzyme in nature and help enzyme to catalyze a biochemical reaction, we call them as a coenzyme or cofactor. So all those cofactor that are organic in nature, we call them coenzymes. So enzyme that are non-functional, we call as the apoenzyme. So apoenzyme is not having any cofactor or coenzyme bound with them. However, the holoenzyme is the fully functional enzyme, and it is having coenzyme or cofactor bound to them. On the basis of their chemical reaction, they catalyze enzyme classified on the 6 different classes. The oxidoreductases are those enzymes, they catalyze the oxidation reduction reaction, and the transferases are those enzymes, they transfer a molecule or atom from one substrate to the other substrate. However, the hydrolases are those enzymes that hydrolyze a bond or release by the addition of a water. And however, the lyases are those enzymes that break down a covalent bond without the addition of water. They mostly release carbon dioxide from a chemical reaction. However, the isomerases are all those enzymes that change the position of the functional group or molecule or atom within a substrate. However, ligases are all those enzymes that join two substrate together, or that make a bond between two substrate. So all this is the basic of the enzyme. Now in coming lecture, we would be studying other important topic related to the enzyme.